changed my talk just a little bit from what was in the title. It used to be predicting traumatic brain injury, and then I decided to add a component to the end of my talk. So now it's biomechanics, a little bit more general, and how a certain structure in the brain, the Pia-Arachnoid complex, really affects the mechanics of the brains and probabilities of injury. So, and I'll explain more about what the Pia-Arachnoid complex is. So let's talk a little bit about traumatic brain injury. It's no surprise that traumatic brain injury is a serious problem, especially at the forefront of the news these days because of all the football players and all the sports-related injuries. It doesn't discriminate with race, gender, age. Anyone can pretty much get a traumatic brain injury. And there is a big need to develop tools that can better diagnose traumatic brain injury and so that we can better prevent or mitigate the severity of traumatic brain injury. So with this need, I come to the table, and several people in my field, with more the engineering perspective. There's definitely a clinical perspective and there's an engineering perspective. And the engineering perspective, especially the mechanical engineering perspective, is that forces are important. And understanding the forces can help us predict injury as well as prevent injury. So there's already a system out there that kind of follows the same engineering perspective. It's called the HIT system. Has anyone here heard of the HIT system? Yeah. So it's basically a series of accelerometers put in a helmet. And then they put them on football players and so they can measure the accelerations of the head, especially during impacts. So football players are willing subjects for traumatic brain injury because they're willing to put this helmet on their head and go run out and play. And oftentimes they may, actually several times they're going to get impacts in the field and they may actually result in a concussion at some point. The challenge with this, and that's what we as mechanical engineers want. We want to relate input forces with the human body's response or the injury response. And the challenge with this data is it's a huge data set that they've been collecting in the US for a while. The challenge is interpreting the data. Okay? So you have a force or acceleration and you know that this player got hit and perhaps he had a concussion or a mild traumatic brain injury, but he probably had 100 hits before that, not necessarily in the same game, but in practice, in previous games. So there's a history that every player has, and every player's history is very unique. So interpreting the resulting data is very, very challenging. And understanding how do we make the correlation where this acceleration is going to cause this injury. And of course, there's one other problem with this. This is a great program because it's going to collect a lot of data. There's one other problem, and that's, do we even know if they're putting the helmets on correctly? Okay? So you get impact, the helmet's moving separate from the head if it's not on right, so then it's not a direct correlate to what the body saw. So instead of doing this direct measurement, one aspect that you can do is computational modeling. And that's really because you can specify a force to apply, and then you can evaluate how the stresses and strains result, how it's the deformation that's going on in the skull. So computational modeling of traumatic brain injury has been around for a long time, especially in adult traumatic brain injury. 1960s, the first model was developed. Anyone want to take a guess on what it looked like? That, okay. So basically a rigid skull, some kind of fluid in the middle. They eventually got more advanced, of course, and then had 2D models, still rigid skull, but maybe some more geometry, more realistic material properties. And eventually we get to today, which one of the probably better well-known models is this Wayne State head injury model. And you can see a lot more detail in it and a lot more anatomically correct features. However, even today, there's several limitations of these models. So if we just take their model, which I'm not picking on their model, it's actually a pretty good model. If we actually look at this, this is one of the limitations of pretty much all head injury models, is the space between the skull and the brain is modeled with single solid elements. Okay? The problem is this is what it looks like in this space. This is what we're coining the term Pia-Arachnoid complex, which is more of an engineering term rather than a clinical term. It's basically encompassing the different layers underneath the skull, so the dura, and then the arachnoid, and then there's these little wispy trabeculae that go below that, the blue there. And of course there's vessels in this space, and there's actually fluid in this space. And then you have something called the pia, and then it attaches to your brain. Okay? So it's actually a pretty complex structure. And as engineers, it's easier for us to simplify those structures. We do not want to model this because it's complex. So we always try to start simple, and then if we need to, we kind of start getting more and more complex. So I would say to date, there's probably no model out there that can accurately predict head injury. They can, estimate, maybe it might, but they all disagree. And actually, predictions of head injury are very model specific. Right? So just like we advanced from simple circle to this kind of model, we need to continue to advance and improve our models. So one of the goals I have is to improve predictions of injury from computational models. And the injury that I'm talking about is axonal injury, which is injury to the the axons in the brain. And the other injury I'm going to talk about is intracranial hemorrhage. So this is hemorrhaging of the vessels, generally on the cortical surface of your brain. So how are we going to do this? How are we going to improve our predictions of injury? And that's really to tie them back to the anatomical structures and start getting a little more complex. So this is a hard thing to do for engineers, but we're going to have to come to terms with the microstructure. Okay? Oftentimes we just want to simplify it into a homogeneous structure, but really it's a little bit more complex. So one of the goals of the research that we've been doing in our lab is trying to see what the microstructure is of the brain and how does that inform the mechanics of the brain. How can we better predict injury with those? So there's really two areas, as I said, that we're focusing on. That's axonal injury. And we're going to be looking at DTI, or diffusion tensor imaging, and correlating the directionality of the axons in the brain with strain fields. And our theory is if we're looking at axonal injury, which are really, axons are long, thin structures, so if you pull on them, you're going to pull in this direction rather than this direction. They're more likely to injure this way. So we're going to look at strain along the primary axis. That's one study. And the other study is looking at that Pia-Arachnoid complex and the microstructures there. I'm really only going to talk about the complex study today. We're actually submitting a paper for the DTI actually next week. So we want to incorporate this kind of microstructure into a model. And we've chosen an animal model. And why did we choose a model of an animal head instead of a human head? Well, that's because we actually have really clear data of how the forces relate to the injury. Whereas in humans, it's not so clear. As I said, there's history involved. You may know forces that someone saw, but you don't know what they saw before. Okay? So that's why we start with an animal computational model first, is because we have the data. And eventually, you want to get back to that human level. Okay, so let's talk a little bit more about the sub-arachnoid complex. So as I said, it's more of an engineering term. You'll hear more sub-arachnoid space. Clinically, it's a series of membranes that separate the brain and the skull. Really, if you think about it, it's the sole mechanical connection between the brain and the skull. So when you get a head impact and your head rotates, your skull is always going to lead, and then the brain is going to follow because these tethers pull it along. Okay? So that kind of begs the question that it's probably an important component to study that probably influences the biomechanics of the brain. So our hypothesis is that a better understanding of the structure and how it affects the brain will improve our computational models and potentially assist in diagnosis of mild TBI, which is very challenging to diagnose and assess and really understand right now. And so those are the two pieces that I'm going to talk about today, is how to improve our models and then also how to assist in the diagnosis of mild TBI. And I added the second part later, so it's a little rougher than the first part. You'll forgive me. Okay. So we actually started this, I don't even know what year this publication is. I think it's 2010. Okay, so maybe 2012, I don't remember. So a few years ago, we actually started this. We thought, okay, everyone's modeling the structure with solid elements. Let's do something a little bit better than that. Let's pick elastic elements. You know, it's kind of stretchy, it should be elastic, sure, why not? And again, trying to simplify the entire problem there. So we took an animal, we developed an animal model. This is of a pig. The red is the skull. We modeled it as a rigid body because we're looking at purely head rotation, no impact. Okay. The greenish material is the brain, and then we also have a phallus in there. And we replaced that layer of solid elements that represents this space with spring elements around it. And we wanted to figure out, how does that affect predictions of brain injury? Does it improve them or does it not? And as I said, we chose the pig model because we actually have associated animal experiments where we know head rotations that these animal heads have experienced, and we know the resulting injuries. So it allows us a chance to validate our model, or at least compare predictions of injury in our model. So we have the computational model. In the animal studies, we actually calculated subdural hemorrhage. So we looked at hemorrhage on the surface of the brain, and we calculated regions of hemorrhage, and came up with a percentage. So you can vaguely see, I don't know if it's clear enough, you can see the different regions outlined, and we looked at how much blood is present in each region. And that was our quantitative way of determining hemorrhage. Okay. So how well did this just changing it from solid elements to spring elements, how well did it do? You probably won't be able to see this, so I'll read it. So on the left here is our spring connector model, and this is our solid element model. Okay? And what we found is overall, the solid element, the previous version of the model, was able to predict hemorrhage with a 70% sensitivity and a 77% specificity. Not that great. Okay? Our spring connector model, so just replacing it with linear elastic springs, improved to about 80% sensitivity and 85% specificity. So it did a better job. 80s is okay. Okay? It's getting closer to getting a B in a class rather than a C. But one thing we noticed with this is if you look regionally at how well it predicted, it didn't do so well. So the old solid element model, it could only predict about 63% sensitivity to maybe 80% specificity, whereas the spring connector model improved that to, in some regions it was 80%, 93%, but in others it was also 63%, 70%. So what that told us is that this model that we decided just to try out was better at predicting hemorrhage generally, overall, that it would be there, but it couldn't tell us where. Okay? It definitely wasn't a great prediction tool. It was better, but not great. So then we had to revisit the problem, right? The whole point was to try to add something to that space and more realistically characterize it. So we had to go back to, let's examine the Pia-Arachnoid complex and really see what it is. We want to know, what are the substructures there? How does it vary? What's the density across the brain? What's the directionality across the brain? What's the geometry of those substructures? What do they even look like? What are the differences within a brain? What's the variability within a brain? And what's the variability across different brains? Okay? We really wanted to understand these structures so that we can better model them. But the problem is how do you do all that? Okay, so we went to the literature. There are some anatomical papers on these structures, but they're more qualitative. They're more looking at, here's an image of what they look like, but no one quantified it, and no one quantified it across the brain. And sometimes they weren't looking in the regions that we were necessarily interested in. They look a lot along the spinal cord, but not necessarily along the cortical surface or the top of the brain. So we had to come up with a way to image these. Typically, you can do it using microscopy, maybe transmission electron microscopy, scanning electron microscopy. You can use a lot of those techniques to image them and get actually really nice images. The problem is you're taking a chunk of the tissue, you're taking it out, you're slicing it really thin, and who knows what you're doing to all these structures when you're doing that. Right? So we really wanted to leave them in the brain. We did not want to cut up our brain. So instead, what we found, I was talking earlier, and I actually have a second research focus, and that's ocular trauma in addition to head trauma. And one of the benefits of that is that I sometimes see tools used in other fields that are not used in my current field. And through my research there, I actually got the chance to become more aware of optical coherence tomography. That's used a lot for ocular research because basically it's an optic system, and the eye is a nice optics system in and of itself. So it's used a lot in ophthalmology. It uses near-infrared light measured backscattering to reconstruct the images, allows high resolution and good penetration depth, at least in the retina. So me being an engineer, and this is what you should all do, is say, I wonder if I put it on the brain, what would I see? And so that's what we did. So we took a system that was not meant for brain tissue and we applied it to the brain tissue. And so this is what we saw. You can actually see what you're looking at these are the gyri of the brain, and this is actually one sulcus of the brain. And the OCT takes a volume, okay, and it takes different scans within the body, different frames within the body. Okay. Here is a video showing the OCT scanning through the brain. You can actually see a vessel on the right-hand side traveling down. You guys see that round vessel traveling down? So we can actually image these microstructures and get a sense. So that told us we could do it. So we did. But this was all done on brains that we took out of a head and we just laid them down and we just looked to see what we could find. Okay? But once we saw that we could actually accomplish this, we decided we needed to get a little bit more systematic about it. So we decided to image it in a very systematic approach where we took a brain, but we actually left it in the skull. In this image, this is one of the earlier brains that we imaged. But we actually left the brain in the skull, so it's in situ, okay? And we imaged it progressively all across the brain to get a really good sense of what's the distribution of these structures. We had to reflect back the skin. We had to do a craniotomy because you can't use the system through bone. We had to reflect the dura, so this is another layer we had to reflect back. We left the dura along the midline, along the sagittal sinus, so that we didn't disrupt the vessels there. And then we imaged the exposed region. What did we do with the images? So we had about 30 regions that we scanned per brain. We took 200 frames for each of those regions, and we had 7 brains, so it's about 42,000 images. We originally tried to do an image processing program, but there's enough noise in here that it became really challenging for us and our skill set, so we decided to let that drop. And what we decided to do is just take a single representative image for each volume, okay, and analyze the volume fraction of arachnoid trabeculae in that image. We validated taking just a single versus all the images. We validated it by actually, in a couple volumes, analyzing the average volume fraction across that volume and comparing it to a single image, and it actually was fairly close, so we felt okay with that. So we're looking at the density of these trabeculae and wispy structures in this region, and that's what I'm going to focus on. All right, so what did we find? Interestingly, we found higher densities of these arachnoid trabeculae in the front and the back of the head compared to the middle. If you looked at the average volume fraction across the brain and compared it with multiple brains, it was only differed by about 4.8%. So the average volume fraction is fairly similar across multiple brains. However, when you looked in a single brain, the difference between the lowest and highest region of volume fraction was more like 20 to 38.4% difference. Okay? So that's a pretty big variability within a brain. And actually, even more interesting is that we found a lot of regions that had high density next to green density. So you see high density next to green density, high density next to low density, where high density is red and low density is green. So you can see in that image that the red and green squares, a lot of times they're right next to each other. So that's a stress concentration waiting to happen, right? It's a pretty big disconnect. So we have these images, and you can see as we moved along that we actually got better and better at doing it and started scanning more and more regions. And that's just how science is with these preliminary studies. So now the question is why is this even important, right? Do we care? Does this variability even matter to brain biomechanics, or is it so insignificant that we just kind of wasted a lot of time? So to evaluate this, we decided to incorporate this variability of volume fraction in our macroscopic model from before. So what we wanted to do is each of those regions has a different volume fraction. What if we somehow account for the difference in those volume fractions in our macroscopic model and then see how that changes our predictions of injury? Okay. So the first thing we wanted to do is come up with a microscale model that simulates these structures, because that's what we're going to couple with our macroscale model. So we took, to create our microscale model, we decided to create a very simplified microscale model. We're not going to model all the geometry of those structures. Again, engineers, we like to simplify things. We like to make our lives a little bit easier. So we decided, let's start with some of the very geometric basic structures that we saw from the images, and let's just start with those. So we have a cord, we have a sheet, we have a longer sheet, and then we have a blood vessel wrapped with arachnoid trabecular material. And we decided we created a MATLAB script that would randomly place these structures onto what we're calling an element, okay? A single element, which represents a chunk of the Pia-Arachnoid complex. And so it's randomly placed, and then you can see the result in 3D here on the bottom. And anytime we did a volume fraction, it was a new randomization process. Okay, so what we resulted with is something that looks like this. We created multiple volume fractions because we wanted a volume fraction for pretty much every region, the average volume fraction. We, of course, did a convergence study. This was the challenging part, of the material properties. Very little material properties are known about these structures. There's definitely some properties about porcelain vessels. There's some properties about the Pia-Arachnoid, actually not that, the dura, which was in our macroscopic model. So we had to estimate what the properties were for the individual structures. And we did that through comparing it to some mechanical tests done on actual whole chunks of this complex. So we had measurements of that chunk in traction, and in tension, pulling it that way. And from that, we kind of estimated some material properties. So no one knows the material properties. We just estimated them as best we could. I will say, now that we have this imaging technique, we can probably measure the material properties on that scale. Okay, so now we have our model, our microscale model. Now we need to couple it to the macroscale model. So the colored regions here are where we have our average densities. So each of those colors is where we applied an average volume, different volume fraction. If it's gray, we just applied the volume fraction of the average of the grain. Okay, so the gray just had its own average volume fraction, and all those other ones had unique individual volume fractions. And we need to somehow implement the mechanics of this microscale model into our macroscale model, and we did that by just assuming a representative element for it. So to come up with the properties for a representative element from our composite microscale structure, we basically did the same thing that Jin did in his actual experimental test. We performed a tensile test across different volume fractions, and we performed a normal traction test across different volume fractions. And our hope was to come up with a transverse isotropic model that we can implement into our macroscale model. And what we found was not too surprising. When we pulled in plane, so this is tension, so I'm taking my model that looks like a sandwich and I'm pulling it this way. Basically, you're just stretching the membranes, right? So it didn't vary all that much with volume fraction. It had a little bit variations based on stress concentrations where they were attaching, but it didn't really vary that much. And then, of course, if you're pulling apart these structures, the number of structures is going to matter, and it related linearly. So for the one direction, the in-plane motion, we used a constant modulus, and for the out-of-plane, it was dependent on the volume fraction of our model. And that's how we just implemented the mechanics of this microscale model into the macroscale, just using this continuum of properties. Okay, so now we've coupled the models. Now let's just see how it does. Okay, so we took our same model from before and our same simulations, and the simulations of the animal experiments, I don't know if I actually said it very clearly, were animal experiments where the head was rotated fairly rapidly, and we have a gradient of injuries, so we're looking at varying levels of hemorrhage, and we also looked in 3 different directions of head rotation. Okay, so we have a lot of data to compare this to see how well it predicts. So just taking one simulation, okay, so that's what we started with. Does this even matter? Are we even going to change anything by putting all this in? Okay, so what you're seeing here is using that connector model, the spring element model. This is the cortical stress. So this is the stress on the surface of the brain with just a single simulation. Okay? And what you see with the connector model is a lot more uniform stress distribution. Okay? The 2 time points are during acceleration, when the head begins, and during deceleration. So we took the volume fraction, the regional variation volume fraction from actually 2 animals, because we wanted to see if we simulate it with this brain, how does it change? And if we simulate it with this brain, how does that affect it? So what you'll see here are the results when we implemented our volume fraction into there. So we're taking into account some anatomical features of the animal, and we're putting it into our model to see what it does. And what you see is a lot more localized stress. This is the same scale bar. So not as blah and out there, it's a lot more localized and focused, which makes sense based on our theory that the connectors could predict overall that the hemorrhage would occur, but it couldn't predict where, right? So this seems to be at least doing a better job of where these stress concentrations may be. But are they accurate? Do we even know if they're accurate? One other point is that there's not much difference between the 2 animals. So remember, there wasn't much difference between average volume fractions between animals, so it doesn't seem like that's playing much of a role here. So again, the question is this even accurate? This is just a comparison of the results. So now let's go ahead and let's go ahead and use this model to predict injury in these animal studies, and let's see how well it does. Remember, the connector model was better than the solid element model. Is this going to even be better than that? Okay. So just looking again at a single simulation to start. Okay. This, what you're seeing here, is the previous connector model. That's the elastic spring model. On the bottom is our predictor of injury. So as our metric for injury increases, we'd expect actually injury to increase, right? So as strain in these connectors, which was the predictor, increases, we'd expect injury to increase. And we saw it for the most part. Each of those dots is a region, okay? And on the y-axis is the hemorrhage, the percent hemorrhage in that region. So for the most part, it does follow a trend, but if you notice that far left upper point, that's 25% injury, or 25% of that area had blood in it, and it should be over with the other ones, right? It should have the same amount of strain in it, but it didn't. And that, again, points to why the model was good at saying there's probably an injury there, but not saying where. When we did the same thing now with our 2 animal simulations, okay? So this is just one animal volume fraction, this is the other. What we found is a much tighter fit. So it actually did correlate with the region. So now we have a much tighter grouping of, if these 3 regions had the same percent of hemorrhage in them, guess what? They also have the same mechanical metric. So that was just a single case that we started with, but it was promising. So we decided, let's simulate all the cases, and let's see how well it does. And that's just what I showed you. Okay, so another table, I know. So what's boxed is the old connector model. Okay? And before I compare the 2, one thing I want to point out is in the previous model, we actually had to have a threshold for the amount of hemorrhage that we could even detect. Okay? So our model wasn't sensitive enough to detect hemorrhage unless there was in each region at least 25%. Okay? That's not a lot. These regions are fairly small, so 25% is probably like that much. So it's decent, but it's not great. The next thing I want you to show, which I have these a little out of order, is the new model could detect 1% hemorrhage. So if only 1% of the region had hemorrhage in it, we could tell that. Okay, so much better resolution. All right, so these are the previous values you've already seen. Overall, it's about 80, 85. Regionally, it was anywhere from 63 to maybe 93. There were a couple hundreds in there, but not great. Whereas there's the new model, okay? And this was the best predictor for this model. Now you see overall, it has about 94 to 100 sensitivity and specificity, so much, much better. And regionally, you see a lot more 90s, a lot more 100s. The lowest is about 86%. So just by incorporating this variability in the volume fraction of arachnoid tuberculate, we can improve our model predictions immensely. Okay, so some conclusions just for this part of the presentation. Incorporating, obviously, the arachnoid tuberculate, it increased the local variability and created these kind of hot spots, which did correlate fairly well to locations of hemorrhage on the surface of the brain. And so our predictions increased by about 15%, and our regional predictions increased by about 20 to 30%. So very vast improvement by just taking into consideration that microstructure and variability. There's a lot of limitations, there always is. We didn't model CSF in our model, and it's fluid flowing through that structure. So that is something to consider, is how incorporating fluid into that structure would affect our predictions. The OCT imaging we did was all post-mortem. If we imaged it while it was in vivo, we still have to reflect all the layers. There's a pulsatility to it, and there's also blood flowing through, which would inhibit some of the imaging of blood. So that's something we want to work out and actually go with more in vivo measurements. The other thing is that eventually we need to go back to a human model, right? That's going to be challenging. I think we could probably image post-mortem at least the variation in humans and across ages of these structures. But then we need the data to validate the model and to determine if our predictions are accurate. And that's the challenge, is knowing if a person comes in with a head injury, exactly what happened in that scenario. And it's always kind of a gray line trying to figure that out. You can approximate it and see if you're in the ballpark, but it's very challenging. And maybe that might be something we can use with the football player data, because then we know exactly what went on. But again, we have a history, so who knows. Okay, so that's the first part of my talk, was how this complex could improve our prediction of our models. We're actually firmly believe that they will, and we've shown that they're actually a vast improvement. There probably is more that we can do. Advancing models is continuing, it's ongoing, it never ends. But now the second part is, how can we use this structure to potentially help diagnose mild traumatic brain injury? Mild traumatic brain injury is very unique in that it's challenging to diagnose. There's no definitive guidelines that this is what defines a mild TBI. It's all based on neurological assessment. And oftentimes when you get a mild traumatic brain injury, if you have a second head injury or second impact to your head, it could result in more severe trauma, even if the magnitude of that impact is lower. And no one really understands exactly why this is. Okay, we know it is, but we don't know exactly why. And so therefore, it's unknown when the coast is clear. When is someone okay to go back to playing football? When is someone okay to resume their regular activities, or are they ever? Are they always going to be more susceptible to a head injury? Okay. So my question is does the PAC, the Pia-Arachnoid complex, play a role in this repetitive mild TBI? Okay. Our hypothesis is that the Pia-Arachnoid is damaged in mild TBI. So you have a mild head rotation. These structures don't seem very strong to me, so perhaps they're damaged. Okay, that's our hypothesis. And then that damage results in a greater vulnerability of the brain because you've lost your tethering. Okay? And so maybe that's why repeated head impacts eventually lead to trauma, or maybe if you've had a severe enough trauma and you get another one, it's even more severe. Okay, so that's the hypothesis. So how are we going to test this hypothesis of the role that this complex has in the biomechanics of the brain? Well, we're going to use an animal model. So this is the same. This is similar to the animal model we used for the validation studies. It's a little bit different. The previous model was more of a severe head injury model. This is a mild traumatic brain injury model. So we developed a custom device to apply a head rotation only to anesthetized piglets, and we chose piglets because primarily we studied pediatric head injury, okay, and so did the previous study. It applies only a single direction of rotation through a 100-degree arc. It's repetitive, it's back and forth. Peak-to-peak velocities are 50 radians per second, and peak acceleration is about 2,000 radians per second. So what does all of this mean in terms of TBI? Here are some of the data. So the y-axis is the percent hemorrhage again, okay? And on the left is the cyclic velocity that we have in our animal model, and this is the hemorrhage from more of a car crash, okay? So more severe. If you get grossed out by images, an image is about to pop up. I'll be quick, but this is what it looks like visually on the brain if you want more of a visual assessment, okay? So a little bit of blood versus a lot of blood. So that tells you it's more of a mild than a severe injury. Okay. So our questions are, does damage to the PAC occur in mild TBI? And how can we even make that relevant to clinically, right? So these structures are so small. So even if they do occur, how is that going to help us diagnose mild TBI? How are we going to bring it back to clinical assessment? And so to do that, we're looking for imaging biomarkers that could indicate that damage has occurred to these substructures. Okay? So focusing on the first one, does damage to the PAC occur in mild TBI? So we have an injury model of mild TBI. Now we need to see if damage has occurred, and we're going to do that using our OCT methodology. Right? We're going to look at the volume fraction, and our hypothesis is that if the animal's been injured, we can look at our volume fractions, and we'll see a reduction in volume fraction. Maybe there's damage, so they won't be there anymore. Okay? So we did this study, and I will say we did it in a limited number of animals because it was a preliminary study for a grant. And what we found was actually a little bit discouraging is that, I don't know, the injury, which is red, the blue, which is control, in some areas, the red was higher than the blue, but the variability was huge, right? And again, remember, in each region within a brain, variability is large. And not only that, we can't do this in the same animal. So this isn't before and after injury, it's a control versus injury, which makes it even harder to assess. So, you know, possibly, but there's probably some questions that still need to be answered. And one of them is, what does even damage to the PAC look like? So we used a volume fraction as our metric. But what if you have something that, an arachnoid tubercular that damages below? Gravity is going to still have a dangle. Right? But what if it breaks the top, then it will fall and it won't be in the volume fraction. Or maybe volume fraction just isn't even the way we want to go. Maybe we need to think of some other parameter, such as like connectivity. So come up with an algorithm to look at the connectivity of the mesh. Does that change? Or directionality. Maybe the directionality of the structures change. So we picked volume fraction, but maybe that isn't even the way to go. So as with most good research, you should, once you've realized that maybe you should take a step back, we decided we'd take a step back, let's figure out what damage looks like. So we're just starting some studies where we're inflating the space, and we're applying known pressure to the space, and we're visualizing using OCT so we can actually measure deformation. And then we can break it to the point that it fails, and we can see what does it look like before and after failure. What does this even look like? And then we can actually come up with some means to accurately determine failure versus damage versus non-damage. Okay? These are still ongoing. And along the side, we can actually look at material properties of these individual structures, right? So if we inflate it with a known pressure and we can measure deformation, we can probably back out some properties of individual structures. Okay, so that's where that part ended. But we still have the second part of our question. Okay? So the first part's ongoing. The second part is there an imaging biomarker? Can we even relate this back clinically? Because if we can't relate it back clinically, it's an interesting science project, but no one's going to be interested in it. Okay, so is there an imaging biomarker to indicate damage to the PAC has occurred? So using our same animal model, we decided to go to MRI for this, to see if we have an injured model, an injured animal. Can we look before the injury and after the injury and see some kind of differences on MRI? And when I was trying to think about what are we going to analyze, I thought, okay, what do the PAC structures do? They tether the brain to the skull. So if they're damaged, perhaps that space, which has fluid flowing in it, opens up a little bit. Okay, so maybe your CSF volume changes. And maybe it doesn't. Maybe it's because it's a pretty well-regulated system. So it was a hypothesis. Let's look at CSF volume and see if that changes. The other was CSF velocity. I thought, well, you have fluid flowing through a channel. You have structures inhibiting the flow. If those structures are no longer there, you might have an increase in velocity. Okay, so we looked at fluid velocity in the CSF space. And the last one was brain skull displacement. Okay, that's the ultimate marker that I would think that would correlate to PAC, because that's the whole premise that I'm thinking about, that if you have a head rotation and there's damage, there should be more relative rotation between the brain and the skull. That one's not as clinically relevant because you're not going to rotate the head in MRI, but it still would be interesting to know to kind of prove how those affect the brain volume. Okay? So these are three markers that we decided to look at. Okay, so the first one to look at CSF volume, we took high resolution T2 laser images and of the brain, and we looked at different regions in the brain and calculated the amount of CSF there. And again, this is the same animal, so we're looking before and after, so it's a lot more correlated response. We also looked at the entire brain, so pretty much the entire cerebrum of the brain. So what do we find? Okay, again, limited number of animals, because a lot of this is preliminary study that we're working on to see what we should pursue. Okay, so what we found, so the red is injury, the blue is before injury, and each pair is a single animal. So we only had 4 animal data. One thing to note is region 1 out of 3 out of the 4 did have a difference, right? We don't want to do statistics on N of 4. I don't think that that's appropriate. However, you can see that there is some kind of difference. So potentially, region 2, not so much. Region 3, not so much. And in fact, here, we have pre-injury, it was all over the place. And region 4, not so much. So the potential's there, but we're not even sure if that's a good way to go. Overall, though, if you look at the comparison, 2 of the 4 animals did have some kind of increase in cerebral CSF volume. So this could be promising. You really would need a lot more animals, because there's probably a lot of variability even in the injury level within an animal, as well as just the anatomical features and what we can resolve. So the next thing we looked at was phase contrast velocity imaging. So it's an MRI technique which visualizes fluid flow, and it's been performed in the ventricles typically. I don't know of anyone who's actually looked at the sub-arachnoid space, really small space. And our imaging guys were just kind of laughing at me when I wanted us to do it, but we did. And so it basically allows you to measure fluid flow by gating it with a cardiac cycle. It's kind of like the pulsatility of it, and then also normalizing it to just basic brain tissue. So the blue is tissue, which should not have any fluid flow. Right? So it's our baseline. And what we saw comparatively, again, this is pre and post, let's go ahead and go to the next slide, was that actually the right side of the brain for the 3 animals that we looked at this with, they did have increases in fluid flow. Okay? Now it's 3 animals, so you can't make any conclusions, but it's possibly hopeful. This one did not, however. Okay? So that leads us to believe that possibly, and maybe if we can improve the resolution, because the challenge we had with this study was the resolution of that small space. Right? How do we improve it? And this is before we actually got a surface coil to get better resolution. Okay, so that was also a possibility, maybe. Okay. The last one was our head rotation. So we used some cinescans, which flash, I don't know what this means, but Chase will. Fast low angle shot allows for very fast measurement times, which is what we need, because what we're doing is in the MRI, what we want to do is we want to rotate the head, not to injurious levels, just like this, okay? But enough that we can look at brain skull motion. And we want to see if before injury, is there less motion than after injury, okay? This has been done in actually human studies where they've laid a person in an MRI, they've had something that lifts their head about, I don't know, maybe a half an inch above, and then the person releases it and their head drops. And they measure displacement that way. Because you're not going to do large head rotations in MRI for space reasons and for the health of the person. I will make a comment that my mentor, once put her head in MRI, we designed a device and she had to do this to measure the same thing. But in order to get the consistency right, I think she had to do it like 60 times in a row. Yeah, and then we had to do it multiple times, so she volunteered for that. Okay, so how do we apply a consistent head rotation in the MRI? We designed a device to actually go in the MRI with a bite plate. The animal's anesthetized and he's mounted to the bite plate, and it basically just applies a nice rotation. It had no electronics on it, right? So it was pneumatically controlled. And what we got from that were videos where then we could track, we could specify certain points to track the motion of the skull and the brain, and then subtract out the two to look at the relative brain skull motion. And the post-processing was done mostly by our imaging analysis guys. Now, one thing you'll note here is because we're applying a rotation, the time factor is very important, right? We need something with a pretty high temporal resolution to capture that deformation. But that limits your spatial resolution. So that's kind of the trade-off here. And you'll see that it's fairly blurry. So we found that the tracking algorithm actually worked pretty well to capture the motion that could detect. There was some error, about 1 to 2 pixel errors due to noise. The sad thing is that our rotation was within the level of our noise. Okay? So the first part you see is just the initial rotation, the head's starting to go. We're not exactly sure why there's a peak there. Okay? So we think it's actually artifact from the motion of the head going. Then you have the impact, which is where we'd like to see the deformation. This is where it stops. In some of the cases, we did see a little peak, okay? But it was kind of within the noise level. And then there's nothing after the end, which is what you'd expect, no motion. So the results of this meant that we need to get we either need to somehow improve our resolution or get some kind of better technique for analyzing these images. And so MRI and image processing is not my forte. So if you guys have any suggestions, let me know. Okay, so some conclusions. Let me see how I'm doing on time. Okay, some conclusions from this is basically damage is still a possible. Damage with the PAC is still a possibility in mild TBI. It can improve our understanding. We need to improve our understanding of damage and increase maybe the level of severity in our device to really make sure we're creating damage. And detecting PAC damage clinically, we have a couple of potential options, but we're always open to more options. Brain skull displacement, I like mechanically, but it may not be so clinically well. So we're still exploring options. If anyone has ideas, we're happy to hear them. I think that says the same thing, so I'm just going to go ahead and conclude because I'm running out of time. So just acknowledgments to all the members in our lab and to our funding sources for the preliminary data, which is from Primary Children's Medical Center. And with that, I'm willing to take questions or comments or recommendations, and please feel free to contact me or check out our website for anything else. Yeah.
So I'm in the field of numerical simulation. So in your abandoned simulation, how the force is applied? Is that an inertial force or?
It's inertial force. So we're simulating the brain rotating about a point.
So the inertial force is uniform or is kind of related to the mass density?
So, well, it's not an inertial force. It's actually a velocity trace. So it's the velocity trace from the head rotation experiments that we did in the animal model. So we know what the velocity of the head rotating is, and we basically apply that same velocity to the skull in our model.
So you have to convert the rotational velocity to the inertial force? Or?
No, we just apply the rotational velocity directly.
Oh, the velocity.
The velocity. Yeah,
so that means you already know the displacement or?
That we're what?
The displacement, the time history of the displacement.
Yes.
Oh, okay.
Yeah. So we have the time history of the angular velocity that we applied, and that's what we're applying.
Okay.
Yeah.
Thank you, Brittany. Very interesting talk. Just curious, have you thought of potentially using Doppler OCT instead of using MRI? I mean, if you started with OCT, but now that would have to be in vivo, obviously,
Right.
and you'd have to remove the skull, etc.
Right.
So,
I have thought about Doppler OCT, and I'll be honest, I'm not very familiar with it. So my understanding, and you correct me, as you know, is it measures mostly velocity, right?
pretty much.
Doppler OCT. So I don't, but you can still backtrack out images from it, because I thought it was more for fluid flow, like blood flow.
No, but I'm talking about Doppler OCT for the fluid
Oh, for
flow
the CSF
in
flow.
that region, because maybe the patterns of the fluid flow could be compared, as opposed to, I mean, you don't have to see the microstructures, but
Right.
you can see the global pattern of the fluid. And if you do a differential kind of, differential assessment, whatever, I think that might sort of give you the aggregate, not conclusion, but the effect from all the microstructural
Right.
damages instead of having to see the whole thing that might be hard to see.
Right. So, I mean, definitely in the images, our resolution is poor. So there's still a lot of error. So that would have definitely a higher resolution. The aggregate, this is the issue with this theory, right? You could have damage to a specific region. And if you're summing everything, you might lose that. But we definitely could use Doppler OCT to look regionally to see if we can see differences in specific regions. I mean, that's one of the challenges. We don't know where the injury is going to happen. We're just trying to find it if it does. But that's an excellent suggestion. Yeah.
Sort of related to that actually, and the ability to maybe do longitudinal studies or in vivo, or at least without removing the scalp. How set are you on using pigs as opposed to, say, rats or mice?
So the challenge with rats and mice is actually creating injury. So it's an inertial injury, so mass matters. And the rat's brain is so small, and I actually don't know if those structures are very different, but rats' brains are very smooth, and pigs and humans have, very gyro and cephalic brains. And so I'd probably be opposed to doing rats, but not any other large animal model that has similar.
The reason I ask, I guess, is I know that, and this is way outside my area of expertise, but there's been some recently published work, actually my sister who did her PhD on this sort of stuff, that they created a window chamber model so that they could do OCT, longitudinal studies, in rats. Like they inserted a window chamber basically in there to allow that, and so that they'd do their injury model and then let them recover, and they could monitor all the way along and so forth. So I was wondering, the only problem is you can't have a window chamber the size of an entire pig scalp, I'm sure. But if you work with something smaller, and I don't know, then maybe guinea pigs or something in between, but that might be a possibility.
Yeah, I mean that's a good suggestion. If we can figure out where injury is, I think actually that might be a good route to go. Because then you could create a focal window right where you know injury is going to be. And maybe there's a way to create injury locally to help, right? Evaluate some of these large longitudinal things instead of this entire rotation model. Thank you. Yeah.
I was wondering if you could separate the cerebrospinal fluid flow rate from the blood flow, such as if a sub-arachnoid artery broke and started gushing blood into the CSF. Would that affect the measurement?
It could. I will say in these animals there was no hemorrhage, actually, in these animals. Because after the study was over, we used nitrogen and did histology on the brain. And that's actually one thing that was interesting is when we developed this animal model, we developed it somewhere else, and then we carried it here. But the injury levels were slightly lower than before. So even though there was a little bit of blood in that initial one, these ones didn't have any. And so we actually want to kind of bring that up. But that would confound it. It probably would confound it if there was hemorrhage. But then hopefully we'd see it if it was significant on the T2 images to see that there's some kind of hemorrhage.
I have another question.
Yeah.
In the pictures of shaken baby syndrome, they often show damage to the frontal and the occipital parts of the brain.
Right.
Is there any interest in sort of zooming in on the frontal and the occipital regions?
So, I mean, we have found, so even in our animal model, we find greater regions of injury actually in the frontal lobe and near the tentorium. In here, we found higher volume fractions in those regions. Now, what the relationship is, I don't know, but we're definitely imaging those regions to try to figure that out.
All right, I think in the interest of time, if anybody has any additional questions for Dr. Coates, maybe you can approach her one-on-one briefly. We'll be here for a few minutes afterwards, but let's thank her again, please, for a wonderful